So hello, everyone. I'm William Poole, postdoc at Caltech, and this is going to be the second workshop for the IWBDA 2021 conference, and it's called From Chemical Reaction Network Compilation to Bayesian Inference. And just a couple of quick sort of logistical things. If you're not part of the Slack channel for this workshop, we'll probably be posting some links there, and it's a good way to we can also use the Zoom chat, but I prefer Slack personally. So if you could join the workshop to Slack channel, that would be great. And maybe Andrey or Ayush, you could post that to the Zoom channel. And yeah, so we're recording this conference. If you have questions, you can ask in the chat, or you can just interrupt me at any time. I'd like it to be more interactive. Without further ado, we can start. So, okay, so let me give a brief outline. We're going to start, it's in two parts, and the first part is on Biocompiler and compiling chemical reaction networks models. So I'm going to start talking about Biocompiler briefly. Then we're going to have a hands-on demo where we're going to use Google Colab to really play with some Biocompiler models and try building some and figure out how that works together. And then we're going to have another sort of back to a little lecture style while Andrey is going to talk about Integrase circuits. And Integrase circuits will be used sort of as a way of highlighting some of the more advanced and powerful functionalities of Biocompiler. And then we can go back to a hands-on demo and play with Integrase circuits or other things. And the point of these demos is really for you to try using the code we have it all set up in an interactive environment, and to ask us, the developers, how to use Biocompiler for what you want. It's a fairly powerful framework, and we want to help you explore it. In part two, in part one, we're basically figuring out how to build large complex models very easily. And part two is about taking large complex models and making them small again, and then connecting them to data. So we're going to start with an introduction from Ayush on model reduction and parameter inference. Then we're going to go on to look at some software called AutoReduce, which is does automated model reduction. So we'll start with a big model and make it smaller. And then we'll use black box parameter inference with Bioscript and MC packages to parameterize these models from data. And again, this is all done via Google Colab, so you should be able to look at the code as we're going through it and try it yourself. So I'm going to start with some background. Underlying all of this entire workshop are chemical reaction networks. And I know that in this audience, most of you probably are familiar with chemical reaction networks, but just to go over what they are in case you need a refresher and to know how we're defining them. We start with a set of species. Here I'm calling them X, but these maybe are things like genes or transcripts or proteins or metabolites. And we have a set of reactions which define how these species interact. So we can write this out in a fancy form where we have some input species, like a gene, which at some rate goes to some output species, like a gene plus a transcript. And that rate is called the propensity. And in general, we allow sort of different propensity functions. Maybe they're mass action, polynomial, or monomials, or maybe they're Hill functions. We're kind of agnostic to what type of propensity function you want to use. And there's two types of dynamics for CRNs. There's deterministic dynamics, where we look at the concentrations of counts. So here you can see that here in red, we're seeing how the concentrations of this reaction network A goes to 2B reversibly change in time. The blue line is A and the red line is B. And you can also have stochastic dynamics, where you look at discrete counts. So that's these sort of lighter traces are showing many stochastic trajectories. Or here are the probability distributions if you simulate this system stochastically. In this talk, we're really interested in deterministic semantics of chemical reaction networks. Although one reason why CRNs are so useful is because you can take the same underlying model, A goes to 2B or any other CRN, and look at it either stochastically or deterministically, depending upon what you're interested in. Everything I'm saying, at least in the first bit on compilation, doesn't really care if you're doing stochastic or deterministic modeling. Which brings us to chemical reaction network compilation. And the first half of this workshop is on the Biocompiler package, pronounced Biocompiler, and really what it is in the name. So bio is because it's biological. Biocompiler is a library of biochemical components and mechanisms. So I won't describe what components and mechanisms are, but I think the names kind of are suggestive. A CRN, of course, I just described, is an expressive model of chemical systems. Compiler is, a computer science idea where you take a simple specification like a programming language to generate something more complex. And so here we're trying to compile chemical reaction networks from something simpler. And the Pi, of course, is for Python. Biocompiler is a software tool that exists to be linked to other tools. And we'll talk about that more later. And I think that's sort of the theme of this workshop is we can start with Biocompiler and link it to build pipelines. And we have a GitHub and a bioRxiv paper, hopefully a real paper soon, if you want to check those out. So what is compilation, and in particular, what is CRN compilation? I think in this audience, we're all familiar that you can take computer code like C++ and compile it to machine code, say binary. And one important idea about compilation is that the same specification can be compiled in multiple ways. So if I write C++ code and I want it to work on Apple OS versus on Windows, I have to use different compilers, compile it in different ways for different chip architectures, et cetera. And so we want that from our CRN compiler. And in fact, I've already talked about one form of compilation, which is chemical reaction networks. So when I write out a chemical reaction network and I get my equations, I can compile the same CRN into two different types of equations, deterministic ODEs or the stochastic Markov chain. And this is a form of low-level compilation where we're going from a low-level description, a chemical reaction network, to some mathematical objects. And again, Biocompiler is going to take an even higher level description to produce a chemical reaction network, which can then be compiled into these equations separately. So this is not really a new idea. The DNA strand displacement community has been using different types of compilers for a long time. So you have these ideas where you can represent a CRN and compile it into a DNA strand implementation. There's also some programming languages that become DNA strands. There's some notions of reaction enumeration, which are sort of frameworks for doing this in a more arbitrary way. And the key with all of these, though, is that they're all about DNA strands. And Biocompiler could do DNA strand displacement. It could be used for DNA strand displacement compilers, but it can also be used for synthetic biology. Another thing that is very similar to Biocompiler is rule-based modeling, such as BioNetGen and Kappa. If you look under the hood of Biocompiler, how it works, it's not too dissimilar conceptually in some ways from these systems, although it's different. Rule-based modeling is very mathematical. Biocompiler is written to be more like a scripting language. So they have a different feel. And finally, there's context-specific compilers that, are focused on some scope. So there's some Integrase circuit compilers. There's some sort of a predecessor to Biocompiler from the Murray lab is the MATLAB toolbox for cell-free systems. And then there's also things like Cello, which are, compiling very simple logic circuits out of very particular parts. So how, that gives you an idea of what compilation is. How does Biocompiler actually work? So I'm going to give you an overview now of how it works before we start trying it, just to set the stage. So Biocompiler is a big object-oriented framework. It's, software, it's complicated, but there's a couple of key objects that we need to know about. There's mixtures, and mixtures represent biochemical and modeling context. For example, cell-free versus in vivo. There's components, these are functional parts, maybe like a genetic construct or a protein. There's mechanisms, and these are models of little sub-CRNs that we're going to use to build a model. And then these things come together to produce species and reactions. And finally, there's parameter classes. There's something called component enumerators, which will be talked about later, but they allow components to produce more components, and global mechanisms are a special type of mechanism. And what you do when you combine all these things with all these complex arrows is you get a compiled chemical reaction network at the end. So let's zoom into mechanisms. I think mechanisms are the core abstraction that makes Biocompiler special. And they're black box CRNs. So you have some sort of function you might imagine where you have a user input and an output. For instance, a gene becomes a transcript. This is the transcription mechanism. But there's many different ways this could happen. I could have sort of a very simple model of transcription, like a gene goes to a gene plus a transcript. Or I could have maybe something more like a Michaelis-Menten model where the gene binds to RNA polymerase and then that complex is transcribed to form a transcript and the process unbinds. Or maybe I want to use Hill functions instead for some reason. Or maybe I want to notice that, there's actually the possibility to have multiple RNA polymerases on one gene, and I want to enumerate all of these different possible combinations and have a even more complicated model, and the list goes on. All of these are transcription, and it would be nice to be able to say, I want my gene, test a model with one particular mechanism, then switch it out to a different mechanism to see if it still works. And that's what Biocompiler is really for. So components are sort of the objects that we build models out of in Biocompiler. So here's an example. This is a component called a DNA construct. And it has sort of subcomponents. So components can be hierarchical. This one has promoters and a CDS and a ribosome binding site and a terminator that we use to specify these different parts. And each of these subcomponents know the mechanisms that they're supposed to be involved in. For instance, a promoter does transcription, and because the promoter knows to transcribe and this component has some smart functionality built into it, you'll get two different RNAs because there's two different promoters in different directions. And the ribosome binding sites tell you that there's going to be some sort of translation happening as well. And what Biocompiler do is it will enumerate a CRN for this model. And it does that by putting it in a defined context, and that context gives you the mechanisms that are going to be used for all of these processes. So we start with all of these components, and we put them in a mixture, which is our context specification. And then we compile that either in a simple context. Here we have a simple model where there's no machinery, there's no ribosomes, no polymerases, but we still have transcription and translation. You can see these are the different DNA states we have that become RNAs and some proteins. Or we compile and say a much more complex mixture. This one has things like ribosomes and RNA polymerase and endonucleases, et cetera. And here you see that there's many more reactions. If you're not familiar with these graphs, these are sort of a bipartite graph representation of a CRN where the circles are species and the boxes are reactions, and boxes connect multi-sets of circles. And Biocompiler produces these graphs automatically. Kind of cool. So let's sort of illustrate this abstraction with an example from biology, which is the Lac Operon. If you're not familiar with the Lac Operon, it is a genetic operon in E. coli that switches or controls when E. coli metabolizes glucose versus lactose. So we're going to have to define a context, the mixture, the parts or components, and the processes or mechanisms needed to do this model. And so we'll start, our context is E. coli, which I'm showing in sort of gray, the Biocompiler class used for this. This is called the TXTL dilution mixture, because it has transcription, translation, and dilution. And just because we know that we're in this context, we know there's transcription, translation, RNA degradation, dilution, and also some pre-built species like RNA polymerases, ribosomes, and RNases. So those are just sort of sitting in the mixture waiting for us. Now we're going to have to add some components. So the first component is the Lac Operon. We're using something called a DNA assembly to model this with a particular type of promoter, a combinatorial conformation promoter. And I'll describe what that does, but these are pre-built classes in the library, and you can also build your own classes. So don't feel like this is the only choices you have. It's very easy to add to this software. And what this is really doing is it's modeling this sort of construct as a bunch of binding sites where different things can bind. And when you start adding in the transcription factors that bind to the Lac Operon, the LAC-R tetramer, which we model as a chemical complex, and the CCRP transcription factor, which is also a chemical complex made of CAMP and CRP. These things allow different states of the Lac Operon to form these different folded conformations where LAC-R basically allows binding in these different exclusive folds, or you have an open conformation, and then CCRP can also bind to these guys. I'm not showing that in this diagram, but they can bind combinatorially. And finally, this is about lactose and glucose. So there's the enzymes that allow lactose and glucose to enter the cell, lactose permease and glucose permease, and they come with the different lactose and glucose species. And you'll notice that we also got more mechanisms with these that we need. We need binding, conformation changes, and catalysis is how we're modeling transport. And beta-galactosidase is finally a metabolic enzyme encoded by the Lac Operon that catalyzes lactose into allolactose. And so when you take this whole thing, we have our mixture, we have all of these parts, we have these mechanisms that we've defined, and they'll sort of automatically include some species. We compile it, and we get a very complicated CRN. So in about 50 lines of code, you can specify this whole thing, and you get a CRN with 173 species and 343 reactions. And here's sort of a schematic of that. So the black box over on the right, the blue square of circles, these are all species representing different open conformations of the Lac Operon where there's no fold, but there can be many different things bound. We can still have Lac repressors bound to different places. It's just not folded. We can have polymerases bound. We can have CCRP bound. So there's a lot of options here. We're enumerating all of these species combinatorically under the hood. And then these smaller sort of groups of species represent the different folded conformations. Because the fold excludes certain binding sites and maybe excludes transcription, like you can't bind a polymerase if you're in these two folded states. You get fewer conformations here. And you'll notice, for instance, that only these two in the model I used, only these two conformations can transcribe. So here the boxes represent mechanisms, and while these two guys cannot transcribe. However, conformation changes are what link all of these different species together. Also, they link them to the different transcription factors. And then down here, these boxes represent different processes, like translation is here after transcription. Here we have binding for all of our chemical complexes. And here we use catalysis to model both transport and metabolism. So this is a way of thinking about a model, not in terms of a CRN, but in terms of the processes that connect different components together. And we can change out how we actually want to model this in terms of specific reactions. So I'm going to give sort of I guess we'll stop here. Does anyone have any questions on how any of this stuff works? Prashant, I see you're raising your hands.
Yeah, so sorry, I don't know if I missed this, but so these are like the inbuilt processes that automatically compile to the CRN, is that correct?
Yeah, so this is a library, so I'm using a bunch of classes that already exist, but you can also build your own classes very easily, just filling out a couple functions.
That was basically my question, because what I was kind of curious about is, what if you wanted to model, let's say, crosstalk, for instance? So is it trivial? Do you just add another class and then that, or is there like another mechanism to define things like that?
Yeah, you'd probably want to add another class either for the mechanism or for the components, or maybe both. And when you subclass component or mechanism, you have to fill out usually two functions to subclass it. We've had freshman bioengineers successfully build their own subclasses for Biocompiler. So it's, I think, trivial to anyone with any programming experience.
All right, cool. Thank you.
So, why would you want to use Biocompiler? It's modular, so you can use it to generate architectures by reusing components. For example, I might start with this DNA assembly I sort of already talked about, a transcriptional unit, and I can wire it together. I can have, say, one DNA assembly wired to another as a repressor and get this sort of inducible repression behavior. I could wire two of them together to get a bistable toggle switch, or I could wire three of them together and get something like a repressilator. So I can take the same piece and explore different topologies this way very easily in different circuits. It's also expressive, so more than just allowing me to explore things modularly, I can explore different ways of implementing circuits. So I could take something like this inducible repression that's very sort of idealized. I can say, well, how am I going to build that in the lab? Maybe I want to use dCas9 and a guide RNA that bind together to get a repressor that binds to this very particular sequence. Okay, well, now I actually have a more complicated implementation. I can still model that in Biocompiler, but of course, it now becomes more complicated because, especially if you include machinery, there's this notion of retroactivity, where maybe if you just build a bunch of guide RNA, you can actually increase the expression of your reporter. So there's some more complicated tuning that has to happen here, and Biocompiler lets you explore those parameter spaces very, very quickly. Similarly, with the transcription factor toggle, this is sort of the standard one where I repress my promoters to make a toggle switch. But you can ask, could I do this at an RNA level? Could I have targeted RNases that repress translation but not transcription. And so here's a model I invented that in fact does this, and this is something you might be able to build in the lab. And finally, Biocompiler helps you, well, not finally, but also Biocompiler helps you manage complexity. So you can test assumptions and simplifications. And I think this is something that is very important when we're building models. It's an art, and it's not always clear what level of detail you need. So if I look at this repress later example, if you take Michael Elowitz's paper and you build a model of that circuit that's equivalent to the paper, it's very, very simple. There's no machinery. Everything's very idealized. You get these oscillations. But if you actually think about what's happening in a cell, we know, for instance, that this here is you can kind of see these DNA pieces that are being transcribed into RNAs that are then being translated into proteins by all of these black dots are ribosomes, where there's many ribosomes potentially on one transcript, and perhaps many polymerases on one gene. And the question is, does that type of effect matter for how oscillations work in something like the repress later? It's not always clear if we can simplify that and neglect all of this complexity. Well, Biocompiler an approach would be, let's just model it. And here's a model with multiple occupancy of polymerases and ribosomes, and it still oscillates, which is suggestive that the simplifications used in the model in the paper are totally reasonable. And I already knew that, but often it's not clear what simplifications are reasonable and which ones aren't. And one way to figure this out is empirically by testing different models. Now, as we're building all these models, you have tons and tons of parameters. It's not clear how to parameterize things. One, I think, very useful feature of Biocompiler is that it uses default parameters. So I can build a model using, I don't know, half a dozen parameters. Maybe they're all values of 1.110, using this parameter defaulting hierarchy, where I can basically use very simple parameter name specifications and reuse parameters across my whole model. And then later, as I say, do literature searches or parameterize my model from data, I can fill in my model with very specific parameters for different mechanisms, components, as well as the sort of underlying parameter needed for that mechanism and component combination. So this is a very flexible way of not having to specify every single individual parameter in your model by hand. You just specify some default ones, and then you go back in later to figure out more detailed ones as you have time. It gets models up and running very fast. And ultimately, this makes Biocompiler very efficient. So if I look at all of the models I just showed you, I think everyone in this room would agree that writing out ODEs is a pain in the butt. So you can see in green the number of terms of ODEs, and it very quickly becomes unwieldy to write ODEs. Even writing out CRNs can be hard. And if you look at the number of reactions in orange or the number of species in blue, say for this multi-occupancy repressor, that is already very unwieldy. We have 180 species and over 300 reactions. You're not going to want to write that out by hand, but we can only write 14 lines of Biocompiler code to compile a model this big. And I think that's really the beauty of this software, is it allows you to compress very, very complex models in very simple specifications and deploy these models very, very quickly. As a reference, I produced all of these models in one day when I was writing the paper for this. So it's not super hard to make these. It doesn't take a lot of time once you know how to use the package. So now we're going to move on to learning how to use the package. But before, I want to see if there's any questions before we move on to a demo using Google Colab. Great, well, let's go open Colab. So if you're not already in the Slack, maybe we can paste these links into the chat. Here we go, great. And thank you, Ayush. And so let's just start with this. Oh, and I want to say, if you want to see some more tutorials, we have a series of YouTube lectures and a lot of example notebooks on GitHub that you should look at. So Colab is here. Please let me know if you're having trouble with Colab. Am I still sharing my screen?
It looks like it's me.
Cool. The little lines went away. All right, so this is the collab. So we can go through this. I'm going to actually just for the, so we have to do some installations. At the top here are links to different useful things if you're trying to learn more about the software package. So the first thing we're going to install is Biocompiler from pip and some dependencies. I've already installed these. If you haven't used Google Colab before, it's fantastic. And it makes stuff very easy, but this does take a few minutes as it downloads. And then we're going to install Andrey Shur's version of DNA Plotlib, which is makes for some nice illustrations later in this. It's not strictly necessary, but I recommend it here. It'll make things easier to understand. Now, as I mentioned, Biocompiler is not a simulator, it's a compiler. It produces systems biology markup language models. So in the Murray lab, we've developed Bioscript, which is an SPML simulator that does a variety of things. So I'm going to download and install that. You could use any SPML simulator with Biocompiler. If you like libRoadrunner or iBiosim or what have you, that should be compatible. We've tested on a bunch of them. So we're trying to build modular software that can be integrated into the frameworks you already have in place. And SPML is a great way to do that. I don't have to tell that to this community, but I definitely think that that's one thing that is sometimes lacking is the ability to reuse these codes together. And we're very interested in integrating this with other people's stuff.
William, can you zoom in a little bit so that it's bigger?
That's a good question. Can I change the size of...
Maybe Control plus.
That will work, but that will be, maybe this will work. Okay, is this better?
Yeah.
So I already showed you a version of this diagram, but what we're going to go through is building these four classes. So we're going to start by defining some components and then some mechanisms. We're going to put them in a mixture and then we're going to compile a chemical reaction network. We're not going to talk about global mechanisms. They're useful, but they're not in the scope of this workshop. And for parameters, we'll show you some of how that works. So the steps for building a biocompiler model is you have to first instantiate your components and mechanisms and add them to a mixture. And here I've included some links to sort of different parts of the library via our examples. If you also look in our paper in the appendix, there's a long list of all of the different things, not all of them, but many of the things in that we've implemented. Biocompiler is a library, so it does take some effort to learn all of the parts and how to use them. You can also just build your own, which I think is a totally acceptable way to use the framework. But why build your own if someone else has already built it for you? But yeah, so as you get started, you'll have to sort of learn some of what these things are and what they exist. And that is, I think, one of the barriers to entry, but I think it's worth it, like any library. And then we'll add parameters and we'll compile and we'll show you how we can work with the compiled models. So I'm going to do some imports. I hope everyone is able to follow along with this. And so here's the first model that we're starting with. So it's an example model where we're going to have a metabolic enzyme expressed from a gene. So my gene is going to be transcribed to produce a transcript. My transcript is going to produce this enzyme E, and E is going to catalyze metabolite 1 becoming metabolite 2. And first, I'm going to just write this out as a chemical reaction network by hand. So Biocompiler under the hood is a chemical reaction network. So you can build species and reactions, and this is how we would do it. So we're defining the species. They have names. Another thing that's common to do in Biocompiler is you don't just give a name, but you might give it a material type. So here I might say metabolite. And the reason why you might want to do this is maybe I have a gene called gene, but I also have a transcript I'm also going to call G. So maybe I call this material type equals DNA, and I say here material type equals RNA, and I still have the same name. And this is just fundamentally all it's doing is it's making strings. And you can see that here. So when we write out the CRN by printing it, you see that we have, DNA G. So this is just a string representation of that. Is going to RNAG, and here RNAG is going to RNAG plus E, and metabolites, etc. And this metabolite M2 isn't called a metabolite, so it's just called M2. So this is just a way of writing a string representation, and it's convenient for keeping track of things. But you could also just use a name and not use material types. That's okay. Another thing we have are called attributes. So if you wanted to say have a phosphorylate or a non-phosphorylate, you can add attributes, which is just a way of appending strings on the end of a name. So that's how you would build it by hand. Now, this is not what we're actually doing. What we want to do is we want to compile it. So let's build the same model, but compile that model. So we're going to start by making some components. And we first make an enzyme. And this is a class that's pre-built. We're going to call it E. And it's going to have substrates and products. And the substrates are M1, it's a list, and the products are M2. And I'm reusing the species I defined up here. And then we make a DNA assembly. This is my transcriptional unit. And it's going to produce a protein, which we're going to take the enzyme's internal species. And this is all, figured out for you, basically. I'm going to define a promoter in RBS. If I didn't give a promoter in RBS, it would assume that you can't transcribe and can't translate. So we're just going to give them names P and RBS. These things ultimately are used for finding parameters when we compile, but for now, that's not important because we're going to use, sorry, I'm jumping ahead of myself. I then have to define my mechanism. So I'm using these built-in mechanisms, basic catalysis, simple transcription, and simple translation. I instantiate them. And these are just the same reactions I showed you before. And basically what these are saying is in the model we're going to do, this is how translation is going to be modeled. It's going to be a transcript goes to a transcript plus a product. And catalysis is just going to be a catalytic second order reaction. And then I'm going to give some parameters. So here I'm just using these default parameters. You can see what they are: catalysis, transcription, and translation. I'm setting them all to 1. If I wanted, say, to have a whole bunch of DNA assemblies together, I could, have different promoters, and I could use more complicated parameter keys that might have the name of the promoter in there. And there's some examples of that in the notebook. So I'm just, for the sake of simplicity, doing things very simply here, but you can have very complicated specific parameters for your whole model. And those can be stored in an external file.
There's a question in the chat, which is, when is .getSpecies used? And basically the answer is that if you have a component, you might want to get what the species of that is. So here we're doing the species of the enzyme itself. So that's like E that we're obtaining up there. Otherwise, if you just put in E there, then I guess it would probably
still work with E. It's pretty smart about these things. But yeah, Andrey answered that question perfectly, which is, when you're defining how components are linked to each other, a protein is a species. You could also put a component in here, I think. We usually want to send in a species. So maybe I could have used this same E up here if I wanted to. For instance, I could do it this way. It's very flexible. It's like a programming language. So I could say, E comp, and then here I could say my enzyme is E, reusing the species up there. And I could also say my protein is the same species here. That would also work. So it's pretty smart at figuring out what you mean. And there's no one right way to do it. I can also often put in string names. So if I wanted to just say this is called E, and this is called E, it might also work. So there's a lot of flexibility here.
It's just that when you do DNA assembly, I think all the things that go in as arguments have to be species, right? Or strings.
I can actually, it will figure out if it's component. It will do the get species automatically.
Oh, okay. Well.
And so now I'm setting my initial concentrations. I'm going to have some amount. So here I'm doing g.dna, right? So if you think about it, this DNA assembly, it really has its internal DNA, but it also is saving some other components. It's saving the protein it produces. It's going to produce a transcript as well that's linked to it. So you can sort of access those if you want. And now I'm going to make a mixture. My mixture, I give it a list of its mechanisms and a list of my components, and I change this to ecomp, and I give it my parameters as a dictionary. Forgot to run this cell. There we go. And now I compile it. So let's do that. And when I compile it, I give it some initial concentrations here, again, as a dictionary. And we can print it, and you get the same reaction we had before. And I want to use this as where we have DNA goes to DNA plus RNA, RNA goes to RNA plus this enzyme E, and it's showing you the rates. And I want to show you this down here just to highlight parameters. Parameters are used sort of a three-part key: the mechanism, the part ID, and the name. And it's going to search for some version of this. So here my mechanism is simple translation. My part ID is RBS. That's the name of the RBS. I gave it up here. And the name of the parameter is KTL. And it's going to go search my parameter dictionary or my parameter file or a combination of parameter files I specify for this thing. And if it doesn't find it, it's going to start defaulting. So the first thing it might default to is simple is RBS KTL, then simple translation KTL, then just KTL. And this allows me to sort of reuse parameters across. Maybe all of my part IDs have the same translation rate, for instance. Or maybe regardless of the mechanism I'm using, if this is my part ID, I want to use this rate. But eventually we just default to the name. And so this allows me to specify a model with very, very few parameters. Namely, I could just use these named parameters or have very detailed specifications for a very complex model. And this makes it very efficient to get models up and running. And it's all printed out. If I say, if I don't want to show the keys, right, I don't get that information. I get a simpler representation. But this is just helpful to figure out how I would say specify detailed parameters. And then I can plot it. So I'm going to simulate it with Bioscript. And NumPy doesn't like something, but that's fine. And it runs. And you can see that, gene remains constant. We get a linear increase in RNA, a quadratic amount of E is being produced, and my metabolites are converted.
We got a question in the chat. Prashant asks, can you define transport reactions, i.e. chemical diffuses into a cell, etc.? Yes, we have compartments as a feature in Biocompiler. I don't know if William goes into detail on that at all, but that is definitely something that exists.
Let me just, while we're on the question of what types of things do we have here, one place to look is examples. So here I'm going to the GitHub. And so we can have CRNs. We have things like enzymes, catalysis, and binding. We have gene expression with transcription and translation. We have gene regulatory networks. We have parameters, goal mechanisms. There's a developer overview if you want to subclass these things. We have integrases, and under specialized components, we have some more things. Membrane models, polymer conformations are what I use for the Lac Operon. There's a whole toolbox on cell extracts that we've re-implemented from another paper. So there's a whole bunch of things, and we would love for people to come in and build more things and add to this code base. It's a library that should be, done by many people. We can only build so much. So yeah, we have automatic visualization using Bokeh. And so here's an example, it's printing out these network plots. And so here you're seeing a reaction, and then it's showing how species are connected together.
Okay, so there's another question. Hatem asks, how can you view the underlying model ODEs? So I think those are defined by the simulator. I think that's how it works.
Yeah,
right?
we actually haven't implemented a way to print out the ODEs. You could load it into SBML if you wanted to see the ODEs. That would be a simple thing to print out, but typically we don't even look at that. We use a simulator that exists. If that's something people have demand for, we could print it out as, say, LaTeX. But as you know, as I pointed out, these models typically have hundreds and hundreds of terms. So looking at the ODE in sort of some sort of form would not be, I think, very helpful for the types of models we're trying to compile. If you have a model that's small enough for you to understand the ODE, you probably don't need Biocompiler. It's when you're trying to build a systems level model with, you know, dozens of parts and components and complex interactions where you probably don't want to be looking at the ODE that Biocompiler really becomes useful. And I think the field of systems and synthetic biology is, you know, rapidly moving towards much larger scale modeling and as a necessity. So to sort of move on, the real power of Biocompiler comes from the fact that I can change my model just by changing my mechanisms. So here I want to use a more complex mechanism like the Michaelis-Menten model of catalysis, or a similar thing for transcription and translation where I first bind to RNA polymerase or a ribosome. Well, I'm just going to rename my mechanisms. So I recreate new mechanisms from these new objects that already exist. And I'm have to define some more species here like ribosomes and RNAP because those are needed for the mechanism. A couple more parameters. You have to have some binding parameters to represent how things bind together. And now I just make a new mixture. I'm using the same components I used above. I can reuse them. Every time you add something to a mixture, it's copied, so it can all be reused. But I'm giving it new versions of the mechanism. So now we're going to have a more complicated model, and I can compile that. Oh, because I changed this to ecomp. Hang on. And now I get a more complicated model with more reactions where I have my binding and unbinding reactions, etc. And we can again plot it, and you'll see that when you plot it, you get a more complicated plot here, showing, you know, how different things go. So let's see, this looks like this reaction is the binding of the metabolite to form a complex, where E is bound to the metabolite M1.
We have another question. Mikhail asks, let's say you made some mistakes in getting to this point. Could you show us what kind of errors will come up and how they hint at a resolution?
That's a great question. So we've spent a lot of time trying to make Biocompiler error messages useful. So let's just give an example. Let's say I forget a parameter. We'll just comment out this KU parameter. Oh, and it's going to come up with a value. And no parameters can be found that match this mechanism, part ID param name equals transcription PKU. Okay, well, the simplest way, it sounds like I need something, but this, I can just know, I know how Biocompiler defaulting works, that I need a KU, and I can put that back. And then it works. So that's sort of an example. You know, the other types of examples, you know, it's all objects, right? So if you wire your objects together in a way that makes sense, things usually work. If I try to say, put in something like that doesn't exist, I'm going to get standard Python errors. Ribosome is not defined. Okay, why is that? Oh, it's because I put a variable up here that's not defined. Okay. Now I could do something like this. Here I'm defining this as a string. Sorry, no, I called my ribo RNAP. That's a problem. Okay, so here it's saying something like, oh, it was expecting a species and I gave it a string. Okay. So I suggest for a lot of the easy errors, you can solve them by reading our error messages. That would be the first answer. If you wire things together wrong to begin with, a good way to debug is to look at these plots, right? Where you can start, or look at the CRNs that print out and read through them and say, is this the CRN I want? Is this working the way I think it should? And these plots can also be very helpful. I think when Andrey shows you the sort of incredibly complex integrase circuits we can build, reading the CRNs is very hard because the strings that represent species become very complex. And instead, it's much easier to look at sort of the built-in graphical renditions that we use. And I'll just note that we can simulate this again, and it works, and we get different dynamics, right? So this graph looks quantitatively different from this graph. It looks, the curves are more complicated. And this is the type of thing that when we get into parameter inference actually can really matter. And that sometimes, you know, we're going to do parameter inference in a very simple model that and it won't match data perfectly. But you could imagine saying, oh, it doesn't match, we need to add more complexity. Well, in Biocompiler, the way you would do that is you just go and change your mechanism. And you won't have to change your whole model. You won't have to rewrite your ODEs. You won't have to rewrite everything. You just go up and say, I want to use a slightly different mechanism for this process. And maybe then things can match data better. So I'm not sure what's our timing like here. This is the end of the first part of the first part. So it would be great if people want to try some things for a few minutes. If you have more questions, I highly recommend you just try writing some code. So an example would be, and we're here to help you, model an enzyme which degrades its own RNA, or model a small metabolic pathway. Or you can go look at some of these example notebooks and see what looks interesting to you, or you can ask me about how would I model this. There's 4 of us here who can help answer questions. If you have, say, a specific modeling case in mind and you want to talk about how you would do that, we could do a little breakout room and discuss that maybe for the next 15 minutes or so. And then afterwards, we'll move on to sort of this Integrase case study, which will sort of highlight some of the reasons why you need these powerful software packages, I think, more than my very simple models, which I know we can all do by hand. Yeah, okay, stochastic models and simulation. Well, the simplest way to do stochastic simulation is, I'm going to just say stochastic equals true. Okay, there we go. We have stochastic simulation. So
but what if I wanted to run it many times and get the average or something?
Then I'd have to write a loop. So again, Biocompiler is not a simulator, it's producing an SPML model. Here I'm using Bioscrape, and we have just sort of Bioscrape as an SPML simulator can do stochastic versus non-stochastic simulation. So I just have passed through this keyword into that package. If you want to use your own simulator that does stochastic and can solve probability distributions and things, go for it. We're giving you the SPML.
Actually, I've got a quick question regarding this. So am I right in understanding that basically how it works right now is you've got the model that's written in Biocompiler that then is converted to SPML, and that's how you simulate it essentially, right?
Yep.
So is it have you encountered use cases, or do you know how much of a overhaul or code change would it be if I wanted to do something, let's say without SPML, for instance, like if I just wanted to have some custom ODE solver or some custom ODE simulation. So I'm kind of curious how easy or difficult that would be, or how much plumbing that would require.
That's an interesting question. So I have thought about things like this. Could you use this framework to plug into different types of simulation engines? And it's not built for that, but one could imagine it's not built for numerics, right? So it's not solving equations for you, it's just producing models. But you could imagine that it puts in a module where it's injecting an ODE into something instead of species and reactions. It would require quite a bit of changing. You keep basically the user interface, but you have to change what's going on under the hood can talk to something that simulates your ODEs for you. So one other piece of software, for example, that I've thought about is this Bavarium software package out of Marcus Covert's lab at Stanford, which they're using for whole cell simulation. And so that is like a multimodal modeling framework where they have CRNs, ODEs, agent-based models, flux balance, all of this stuff. And I think it would be possible to take something like Biocompiler and have it take a large pre-built, predefined simulation and use it as an API to access that simulation. But that would be a lot of work under the hood, because again, we're compiling SBML at this point. Marcus Covert at Stanford, Bavarium. Here, I'll send you a link. Yeah, we should get Iran, who is the lead author on this, to come talk at IWBDA one of these, maybe next year. I think there's some very cool things happening in the systems biology world. All right, well, it sounds like we don't have too many more questions. Maybe I'll hand this over to Andrey, who can talk about integrases and yeah, how they work. And I think this will hopefully highlight why we need tools like Biocompiler and what it was really built for. It was built to deal with, in serious biological complexity.
Okay, how do I get my presentation set up here?
You have to share screen, which means I need to figure out how to stop sharing my screen.
Okay.
For all of the things I know about this stuff, I still can't use Zoom.
All right, let's see here.
Here we go. Oh, and stop share.
So use the PDF. Which is somewhere. I thought we were going to combine the presentations. I guess we didn't do that.
While Andrey is doing this, I do want to reiterate that we would love to collaborate with more people and connect this to all of your tools. There's many people doing really awesome things in this room right now. And so please talk to me offline or any of us about how we can extend this further. I think there's lots of room for using this and applying it.
Actually, while that's loading, I've got another question about this.
Please, we're here to answer questions.
So I'm curious if the tool, if you've either thought about it or if you have any case studies, something where you integrate, let's say, Biocompiler with all these public libraries, like for instance, let's say path-based libraries, for instance, like KEGG or Reactor or something. So is there some kind of or if you've actually encountered any use cases where folks have used Biocompiler for that? So I'm just a bit curious about that in general.
What public libraries are you talking about? I didn't know those two you mentioned offhand.
Sorry, so these are basically pathway libraries like Reactome or KEGG, but in general, I'm just kind of curious if you've had any use cases where you connect to these, like you can basically get like either a small or a giant network, and then convert that into a model using Biocompiler.
Got you. Yeah. So we had a summer undergraduate last year, not working with any of us in this room, but she took, I think, a flux balance model and converted it to a Biocompiler model, for example. I think that might have been a big model. So certainly we've done things like that, and it was quite easy to do. So yes, we've just started that on the surface. I think a big problem with that type of approach in general is that there's not often kinetic parameters in a lot of these models, especially the flux balance ones, or they're not very good if there are kinetic parameters. So that maybe will bring us into the last part of this presentation, is how do you connect the stuff to data after Andrey is done.
All right. There's a question. How to generate S-ball visualizations for genetic designs using Biocompiler? Well, I can talk about that.
Yeah, you'll see a bunch of that here. So good question.
Okay. All right. Should I do it? Should I go?
Yeah, go for it.
Okay, great. So I'm going to talk about Integrase Reactions in Biocompiler. And first, I guess I want to start to explain what is an integrase. Why would you want to use it? Who cares about it? So if you guys know about what bacteriophages are, they're basically viruses that infect bacteria. This is a standard diagram of the way that they work. So they can infect the cells. They can integrate their genome into the bacterial genome, and then when the time is right, the genome pops back out, and then it makes more viruses. So this is the difference between the lysogenic cycle and the lytic cycle. Integrases are pivotal in the integration part, and there's also something called excisionase, which I won't talk about, which basically reverses the process. But the basic concept is that a bacteriophage, which has a genome made out of DNA, uses this protein called an integrase to stick its DNA into the genome of a bacterium. And so many other viruses also use integrases for the same purpose, but using different mechanisms. The thing that's cool about bacteriophage integrases is that they are small, self-contained proteins. All right, I had to turn off my sound for the outdoors there. Okay, so yeah, the thing that's cool about integrases is that this is what they're used for in nature. This is what they evolved to do. If you look at the mechanism, oh wait, sorry. So the cool thing is that they are sequence specific. So you can have these things called attachment sites. And for synthetic biologists, you know, what we're doing a lot of the time is we're having a piece of DNA that has a circuit on it, and you know the order of bases on that DNA determines what the circuit does. So you know the ability to change the order of those bases to reconfigure that piece of DNA as the reaction is running just gives you another sort of angle to modulate your circuit activity. So you can, you know, repress or activate genes, but you can also change what those genes are. And that's what integrases really kind of allow you to do. And so here I made a list of like all the possible reactions that two integrase sites can do. So if you have integrase sites that are flanking a gene, you can flip its direction, you can cause a gene to get deleted. And these are all the different things that basically using a serine integrase, which is the type that you find in bacteriophages, allows you to do, because it has these directional sites that are sequence specific. And once the reaction is done, that is to say, once the sites have reacted, it doesn't go backwards. So that's another thing that's really useful for the purposes of synthetic biology. For engineering, you know that this reaction is going to go in one direction and not the other way. So that's one of the reasons why people use integrases. For whatever it is that you know you think that would be useful, where you can, you know, change the sequence of your constructs. If we look at the mechanism of the integrase, which is you know, as William mentioned, the mechanism is the key to Biocompiler. You can see that, you know, what you get is you have these two sites that have to get bound together with 4 copies of the same enzyme. Then what happens is that the sites, the DNA is cleaved, the sites are rotated around, and then you get these recombined versions of the same sites afterwards. So this is the serine integrase mechanism, which is what I'm going to be focusing on, because this is the type of integrase that a lot of people use for synthetic biology. And the good thing about it is that it's one enzyme, it's pretty small. The sequence that the integrase binds is pretty small, it's around 50 base pairs, and the reaction happens pretty quickly. So within, like, you know, 60 minutes here, you can already see that most everything is integrated. It's just not super important what the data is, but it's a quickly acting enzyme that is useful, basically. And like, you know, just because of the fact that this enzyme is very sequence specific and well defined, you can do stuff like think about the composition of these binding sites and how that can lead to producing circuits that can have different DNA states as their output. So not just the concentration of proteins as the output of your circuit, but also just the configuration of your DNA itself can be the output. And so here's an example from our lab for if you can nest integrase sites in some kind of configuration, and because the integrase sites are nested and the reactions that they can undergo have it sort of ordered. Well, they can happen in an order, and depending on the order in which you trigger the integrases, a different DNA state is produced. And so this is an example of an event recorder. You can, you know, depending on which integrase is activated first, you lead to either GFP or RFP being expressed, which is pretty cool. Just one, you know, example of something that you can make with integrases, as William mentioned, there are many papers out there where they try to make compilers out of integrases to do any kind of logic this way. So that's what, you know, to me makes this kind of functionality useful. Any questions so far on this? So there's a chat, but it is old. Okay. Cool. So yeah, given this is something that's interesting to us, you know, it does present problems from the point of view of modeling, because you know, what William mentioned was that, you know, you define your DNA assembly, and then that leads to what all the reactions are created in the model. But the problem with integrases, that's basically kind of like the top drawing over here. So yeah, so these triangles, as I mentioned before, are the integrase sites, and you can imagine. Let's see, can I annotate this here? Yeah. Okay. So like this part is basically what we talked about already. So this is like, you know, a DNA construct, a DNA assembly, DNA construct, DNA assembly are slightly different objects in the program, but you know, the idea is that you have a piece of DNA that makes a piece of RNA and that makes a protein, right? So that's what we talked about already. But the thing that integrases let you do is they let you sort of, you know, flip the DNA in here, so you can flip the promoter over. So now not only is your DNA making RNA, making protein, but it also has the ability to make another DNA through the action of the integrase. So you can make, you know, this DNA construct, and now the promoter is flipped in the other direction, and the only RNA that you make is this thing, which doesn't make a protein. So you can either have a state of your DNA which leads to protein production, or one that doesn't. And maybe that's something that you're interested in. Maybe that is, you know, the change between this species and this one is sort of the thing that you're reading out in your experiment by looking at how much GFP is made. And so that's the kind of thing that, you know, the Integrase Reactions combinatorial compilation allows you to do. That's what I'm going to go into next. So on the right here, you can see that I have made this schematic diagram of the reaction networks present here, and in yellow are indicated the integrase reactions. So you can see that, you know, a piece of DNA can go through an integrase reaction to go to a different piece of DNA. So that's what the white circles are, they're DNAs. When the DNAs get bound by things, for example, RNA polymerase can bound to the promoter, then they get turned into these gray circles. So this is like the promoter bound version. And so that presents another kind of problem that we've solved in the code, which is that if you have if you have this promoter bound to RNA polymerase, there's nothing really stopping the integrase from still being active here. So, you know, the combinatorial nature of this means that, so, you know, in this configuration, you can imagine this DNA turns into that one. But if the RNA polymerase is bound, that shouldn't really affect what the integrase sites do. And it doesn't in the code, because you can easily just flip the promoter with the RNA polymerase still bound. And that leads to the proper reactions being formed there. So that's just a mini example of why, you know, component enumeration is important, because now not only do you have a piece of DNA that's producing another piece of DNA, but you also have, you know, all these other sort of alternative states of the DNA, such as bound to RNA polymerase, being made into the proper version of them when they're flipped into the other piece of DNA. So yeah, any questions on that so far? No, okay. Yeah, so you can imagine doing some really complicated stuff. So if you have a plasmid that can react with itself, you can imagine any number of, recombined versions of that same plasmid that you could form, given that you can take any at B site and any at P site combined together. Those are the two different sites that combine to make the integrase reaction happen. Let's see, there was a chat here. Okay, thank you. Yeah, so, given that you can have all this complexity, you can, the thing about this is that, okay, so you started out with this construct over here that has 2 integrase sites. And then at the end, after you've recombined it some number of times, you still have, 2 active integrase sites. So essentially from the point of view of, can this recombined version of the plasmid still react? Absolutely it can. And so at that point, it's like, this is where you make the choice of how deeply do you want to simulate this? Do you care about this recombined version of the same plasmid? That's something that you can specify. How deeply do you want to recurse here? Because you can recurse infinitely and create infinite DNA constructs. Just in this relatively simple example of like the flipping promoter reacting with itself in the cell, which is something that could actually happen.
Andrey, can I interject for a second?
Yeah.
So for those of you who maybe are familiar with polymer reaction networks versus chemical reaction networks, what Andrey is getting at is that integrases are an example of an infinite chemical reaction network, sometimes called a polymer reaction network. And he's about to tell you that Biocompiler is designed to use recursion to enumerate those up to a certain depth.
That's right. And so, William showed you this diagram, and the thing that I want to highlight in the diagram is that, you can have this component, right, which, a DNA construct is a type of component which leads to a component enumerator, and that can go back and create more components. So, if you need to, you can have a DNA construct that makes more DNA constructs, just like I said, as a function of what the integrase does to the DNA construct. And the integrases, of course, are also represented in the form of being component enumerators.
There's a question in chat.
Yeah.
Asking.
So how can you encode logic? So I guess my question is, what do you mean by logic? You mean like how can I make the yes? Well, so in the the logic gates, I mean, so here's the thing. What Biocompiler does is, given this specification of a construct where you told it that you have these pieces of DNA in this order, in a string, and then also the fact that they're circular, you specify that. And what Biocompiler does is it tells you that, okay, these are all of the products that you could get from having this, initial DNA construct. The logic, is up to you, right? It's like, that's the thing that you come up with when you make your construct, right? So we don't make any sort of, we don't try to do any sort of logical compilation when it comes to integrases. The whole point is that the compilation will tell you what will happen, right? And so, it's up to you what you do with that information, I guess. Yeah, so, yeah, I mean, I think that, yeah, if you were trying to design logic with integrases, then I think that you would consider doing a higher level sort of, compilation. I mean, yeah, so what Biocompiler does is it goes from this sort of, I guess, gene specification type of level to the lowest level, which is, chemical reaction networks. And so if you wanted to go to an even higher level, which was logic to, DNA construct design, I think that would be a level that we are not capable of doing. Yeah. Right. Okay. And so, just to going a little bit deeper on this, this is a little schematic of a little bit more detail of how the code works. you have this thing called a global component enumerator, which tells you, which is how the integrases get inputted into the mixture. So in your mixture, you have components, which in this case are DNA construct type components, and you have a thing called a global component enumerator, and that determines how the integrase will work. So you can define different integrase mechanisms, and by that I mean like how many attachment sites there are, can they react with each other, what are the products of the attachment site reactions. As I mentioned, at the beginning, that this is the let's see if I can go back here. This is the mechanism for serine integrases, but maybe you have a different one in mind, and that's something you could do with the help of defining these global component enumerators. Anyway, once you have a global component enumerator, it will be able to go through all the DNA constructs in your mixture and identify what new DNA constructs can be made. And that leads to, first of all, defining, what is the nature of these different constructs that can be made, and also what reactions lead to them, what specific integrase recombination reactions are required, and that is something that gets put into the CRN at the end. Okay, I'm just checking the chat here. Yeah, so this is just an overview of how that works. Basically another thing that I wanted to point out here, so in this schematic, I'm not really simulating the binding of the integrase. As I mentioned, the integrase is a tetramer, so it binds dimers of integrase bind to each site, and then that binds together to make this tetramer. In this simulation, I didn't specify that because what you can see here is that there's just one reaction that goes straight from the DNA to another DNA. But binding is something that's useful if you care about how much integrase is present. So you can specify that. And when you do, the same kind of reaction turns into something more complicated, as you can imagine. Once you start specifying binding, you end up with these conditions where you can have different states of the DNA. Like in this case, you have only the P site bound or only the B site bound, and only when you have the two of them bound can the integrase actually recombine to flip the promoter. And that's why you only still see these two yellow reaction pathways that are the integrase reactions, even though there's all these new binding states that are now possible now that I said that binding is possible. Let's see, there's a chat. Is there a paper that showed some experimental validation? So there are many papers that talk about integrase functionality. I mean, yeah, I think there are many papers out there about this mechanism and how this works. I think the original Bioscript paper has experimental validation of an integrase circuit in it. That was before we wrote this compilation thing, but I think the idea is, I mean, the experimental validation is there. So
we'll show you an experimental case in the next part of this talk where we actually look at this very simple switching and look at some data and model it. And that will be sort of the simplest case, and that's from a paper that is on bioRxiv and we haven't gotten around to publishing yet. But this is very well studied. If you Google integrases, you're going to find tons and tons and tons of things and experiments. Fewer simulations though, because as Andrey is pointing out, these things are incredibly hard to simulate. They can do so many things.
Yeah, and the thing is that, if you're simulating an integrase circuit, what a lot of people simulate is just what I would call the simplest case. So in this case, in the schematic that I've shown here, there's only the ability of the promoter to flip. You don't really simulate the ability of plasmids to interact with other copies of themselves, because that's too complicated. And I think in a lot of circuits, people try to avoid that. But with the software that we've come up with here, there's no reason why you couldn't simulate that, and maybe that's something that would be useful to whatever circuit that you are interested in constructing. Okay, and so I think that's the end of my presentation here, which means that we can move on to the Colab notebook, where there are examples of how to put integrases there. So I'll give people like 5-10 minutes to reconfigure there while I do that as well.
Let me can we move to the collab pretty quick and take a break in a few minutes? Is that okay, Andrey?
Okay, yeah, sure.
So I just want to make a couple comments about what Andrey just said in terms of what I said, is he was, showing integrases as this, they're very complicated, right? You start with one simple genetic construct and it can produce an infinite number of genetic constructs. And the component enumeration step of Biocompiler is a sort of a black box. You can put any code you want there that can do things like this, but it plays nicely with all of these CRNs. So it's a way of fairly easily defining very, very complex networks from simple specifications. In this case, you start with, say, a specification like you're seeing here, which is could actually be an infinite CRN, right? So that doesn't take much to need this type of functionality to understand biological systems. And what we've tried to do is build a framework where you can take, okay, I want to make subclass component enumerator. I can write any code, and it just will plug right in with one function that tells it how to communicate with the rest of Biocompiler. So you can then produce CRNs and integrate models and produce SBML models in very easy ways.
Yeah, and so I haven't actually run the notebook on my computer, so I have to do that to install everything. So it'll take a little bit. But in the meantime, I can talk about the way in which DNA construct is used, which is the class that I use to specify orders of DNA, like a linear ordering of DNA parts. So here,
can you make the font a bit bigger? Control+.
Yeah, actually, you know what I want to do is I want to make this window smaller, so it's less of a rectangle. Let me try to reshare it and only share the window. We'll see if that works. I have one of these, wide monitors. It's the curse of the wide monitor. Okay, let's see if this works. I'm going to try to share just the window. Is that better?
Yep.
Okay, cool. Okay, so no, please no. Okay, so the thing that I'm doing here is I'm specifying components, the parts of the DNA that are going to go into my construct here. So you can specify this as a promoter that has a name, and a promoter is, a component that has built-in stuff like transcription mechanisms, all kinds of stuff like that. So that's, what a promoter does. You can specify ribosome binding site, which again, comes with the ability to look up parameters under its name, as well as, storing the translation and stuff like that. And the idea that you can specify a coding sequence that makes GFP. This is essentially the same as saying there's a GFP protein, but a protein is a little bit different from a piece of DNA that encodes a protein, so that's why this exists. I'm also setting an attribute here, and the thing with Integrase reactions is that sometimes you want to try to control the complexity, as William was saying. If you allow infinite recursion, you can get infinite different DNA constructs, which is not necessarily something you might want if you're just looking at, let's say, promoter flipping. And so what this attribute does, so this is any part on your piece of DNA can have this attribute, and what it does is it just prevents the plasmid from reacting with copies of itself. So now you're looking at, for example, let's say that you were modeling something on the genome, there's no other copy, it's just a single copy, and you don't have to worry about it reacting with another copy of itself. So that's what this attribute is doing. Okay, and then of course, the whole point if you're trying to model integrases is you want to specify an integrase site. So here I've called it @p, and it's called an integrase site. This is its name. There's also something called a type. And so a site can have a name that's different from its type. So for example, if you want to specify unique sites that have the same type, can participate in the same integrase reactions, but one of them is bound by something else, for example, you could do it that way. And here you say what the integrase is. that reacts to this site. Again, you know, the way that this works is it works on the DNA level. So things are a little bit different from how you would encode, let's say, a promoter that makes an RNA or something like that. Let's see, can I see the chat still in this configuration? Where is my, okay, there's no chat. Okay, good. Cool. So then you can specify your attachment site this way. You say what integrase it binds to. Importantly, you also say whether you want to model binding or not. And that's, again, you know, just this sort of simplified version of Integrase Reactions, which is just DNA goes to a recombined DNA, or if you actually want to model the tetramerization and stuff. That's what that does. And so here I'm adding a mechanism to the attachment site, and this is something that you normally, this is something that you have to do if you say that you don't want to model binding. And so an Integrase site normally has a, and then you can see, so we have like little descriptions on these. An Integrase site normally has a type of mechanism called integration. So here what I'm doing is I'm replacing that with a simpler mechanism. And so the normal integration mechanism only works between sites that have the bound integrase, but since we're not modeling binding, we have to have a simpler integration mechanism, which is what this does. So that's how you do that. You have another Integrase site. But so far, up until this point, we haven't really defined what the integrase actually does, which is to say that it takes an @B and @P site and turns it into HTL and HTR sites. And that's something that's specified in later part of the notebook, which should be in here somewhere.
Just keep going down, Andrey. I
Yeah.
just added, you just hit enter.
Yeah, I hit enter on this, yeah. Okay.
And now,
oh yeah, so what this does, right, so of course what we've done up here is we've defined the parts, but we haven't really put together what order they're in, right? And so that's what this last line over here does. So what this does is it tells you what order your DNA parts are in your DNA construct. So, you know, this is like when you draw your vector diagram, you're like, this is where the promoter goes, this is where the RBS goes. Essentially, that's what this is. And you can see here that I have my @P site, I have my constitutive promoter, I have my reverse @B site, and then I have my RBS and my CDS there. And I've said that the construct is circular. This is important in the case of integrases because when you recombine things, it's important to know if they should be circular, or if they should be cut into 2 pieces. That's the kind of stuff that's important there. So now that you've made this DNA construct, so back to the question of how do I, you know, do the S-ball representation? Well, it's like this. So this is our construct here. It has the attachment sites forward, attachment sites reverse. If you want to not have it reverse, here I'm just going to change this real quick. So now they'll all be in the forward orientation. So there we go. I flipped this around so it's in the forward orientation now. And the difference between, you know, having them on different orientations just determines, you know, what kind of integrase reactions are going to be possible with this piece of DNA. Like I mentioned in the presentation, there's all these different possible reactions that you can get, and it depends on the orientation and the position of these sites. Okay, so here we have, you know, messed up construct here. But we haven't actually, up to this point, we haven't said what the integrase does. And so that's what we're going to specify here using this thing called an Integrase Rule. And the Integrase Rule, what it does is it says that you have these are the types of the attachment sites that come together to make this type of attachment site. And likewise, the same thing happens for ATTR. So this is just a serine integrase mechanism. This is what they're called in the literature. You can call them whatever you want, but it has to match up to what it says up here. So this is the type field, and if you wanted to call it something else, you could change it in here, and then you'd have to change it up here as well. Otherwise, it's like you have, you know, a compatible integrase site in your construct, which you can also have. So that's how you do the integrase rule. This is just saying this is what the integrase does. Then you have to define an integrase enumerator. And this is just saying that this is the name of the integrase, right? And the mechanism that it uses, that is to say the rule, is this. So now you're saying that this is the name of the enumerator. It uses this integrase, or it defines the mechanism of action of this integrase. So if you wanted to include another integrase in your construct, then you could imagine doing something like defining another integrase here and saying that this is like TP901. That's another one. You could indicate that as the name here. And importantly, you would change it in here. But actually, you need to redefine the mechanism as well. That is to say, the rule of how it works, which is basically the same, except here it would say TP901. So then, what it would look for is if you wanted to make integrase sites that work with this integrase, you have to go back up here and change which integrase the site works with, right? Or up here, I guess. So you'd have to change this to TP901, just as an example of adding another integrase to the system there.
So the point you're making, Andrey, is that this entire system is completely modular and can compile sort of arbitrary complexity. Yeah,
that's right. So yeah, if you can redefine what your integrases do, make them, change how they react with the sites, all this kind of stuff. Let's see, I'm going to get rid of this because I think I had the same name there. That's going to screw stuff up. Okay, so then now that you've done that, you have to define your parameters. And the thing that you have to do with the parameters is you have to have this k-int. This is the rate at which the integrase acts, the rate at which the integrase recombines the DNA. This is the parameter that you need for the integrase to work. And after that, it's basically the same as above, except you are adding a global component enumerator here. And that global component enumerator is the integrase enumerator, which has been defined up here. So if you wanted to add another integrase to your system, you could do it like that, assuming you defined it up here, which I didn't. So yeah, right now, basically what this is saying is everything that goes into this mixture, and that includes the integrase construct that I made. So that's the thing that we defined up here, this thing. That is going to be acted upon by our integrase, which is defined by the global component enumerator. And as I said before, you can have arbitrary depth to this enumeration, and so that's what you're defining here, which is the recursion depth. So now you're saying you're only going to really do 3 cycles of this back and forth thing here, where you do. I don't actually know if my mouse is visible on this. This type of window display. Yeah, so you can you can say how many times you want this cycle to be completed. Essentially, that's what this recursion depth is.
And can I just make a comment? You switch the order of your integrase. Maybe you should go back and reverse it so we don't get a giant circuit in a second when you hit compile.
Yeah, I actually think it won't make. Let's just try it just for a second here, because what we've done is we've said that the plasmid doesn't react with itself. So that means that all you're going to get is the intramolecular reaction. So here you have your original plasmid that I specified here. What can happen now that I reverse the site, is that you can have the promoter get deleted.
That's a good question. So maybe component enumeration is another level on top of components and mechanisms. So basically, I mentioned these 3 main classes initially: mixtures, components, and mechanisms. Component enumerators are a 4th class that can be used by components to create more components. So it's just part of the compilation framework, and it's necessary in order to do things like integrases. So it's distinct from mechanisms and components, but it plays nicely with mechanisms and components. So once you get these components, that you enumerate, they can then use mechanisms, et cetera, as if you had built them by hand.
Yeah, so just to review what I just said here, what we have done is we have defined parts of a DNA construct, and these parts within themselves have our components, and they have within themselves knowledge. For example, a promoter knows about transcription and stuff. RBS knows about translation. An integrase site knows about integrases. And so basically we define the parts that are going to go into the DNA construct, and then we define the construct itself, which is an ordering of parts. And this construct, what it does is it will go through the parts and it will basically enumerate these components automatically. And this is going back to the component enumerator thing. It's kind of something that's like hidden away within classes such as DNA constructs. So this already knows how you want to enumerate your components, because it takes into account the idea that you could have promoters and terminators and RBSs and stuff like that. And that's how, using that logic, it will create components that then lead to the production of species and reactions later that are correct. So, like, for example, if you wanted to define a new way of order, like a new central dogma that you could have, and then, like, let's say a promoter would read in both directions now instead of just forwards, you would have to change that in the DNA construct class itself, which would be, just double checking the chat there. So yeah, you define your parts, you define the ordering of the parts. DNA construct already knows about things like the fact that it's, what you're talking about is parts made out of DNA. What you're talking about is things that have promoters. What you're talking about is things that can make RNA. That's already inherent in the idea of a DNA construct as a class. Okay, so now that you've done that, then you can apply things like global component enumerators, which take DNA constructs and figure out how to make new DNA constructs. And that's what we've done here, is that we've started with this DNA construct, which has our ordering of parts, and then made these two new DNA constructs that have new orderings of parts based on the global component enumerator, which is our Integrase enumerator. And you can have different types of enumerators. I think this is the only one that we have defined in the library thus far, but you can imagine doing something like RNA splicing, etc. Let's see, I have a chat here. No, it's okay. It's not a question. Great. So, so now that these DNA constructs are here, not only are they automatically defined based on the presence of integrase sites, because we have our Integrase global component enumerator, they also have a knowledge of what reactions lead to the production of each of these species. Like this, not only is it a DNA construct, which is to say a list of components that has internal logic as well about how they are supposed to interact, it also represents a species that's going to be in our CRN, which is this piece of DNA. And all of that is encapsulated in the fact that it's a component that is in the mixture, right? So this is our mixture, this is our component, and it has the ability, it is a species as well as it knows that it can produce other species. Okay. So now what we're doing here is we're going to make a reaction diagram. This is an old one that I'm going to regenerate now. These are all the different colors that we're going to color the little dots with. You can look at I'm not going to go into too much detail about how that works.
So to show this plot, because we have a question, Andre, so show this plot and I think we'll make some comments on it. So what Andre just did is he's plotting the compiled CRN. So he was saying that you can have these components that are being enumerated, and then we compile them into a CRN. Here's an example, and he's going to show you this very cool interactive visualization, which is going to show you how these components, not only do we enumerate what integrases, what integrases do at the sort of level of here's the different pieces of DNA that will be present, we can then compile the reactions that produce those pieces and how they bind to things and transcribed and translated, et cetera. So all of this plays nicely together. Component enumeration is a top level thing that produces components, and then we compile that into species and reactions, which can be a very detailed graph.
Yeah, and so here, so what we're doing here is we're doing this representation, like William said, which is CRN representation where the species are circles and the reactions are squares. And so there are also these edges which are drawn here that represent the logical flow of all this stuff. So for example, here's a piece of DNA. This, actually, wait, let's start with this one. This is the construct that we started with. This construct has all of our sites in the order that we want. And if you click on it, you can see all of these reactions that it's involved with. One of them is an integrase reaction. So here you have this piece of DNA. Here's our BXP1 integrase. It's involved with this reaction as well. And what you can see if you click on this reaction is that the outputs of the reaction, so there's an arrow going in here, and there's arrows going out into the two different new DNA constructs that are made when the integrase acts on this one. So this one results in the deletion of the promoter in between the sites, and that's just a function of the fact that they are pointing in the same direction. And you get this as one of the products, and the other product is this. And the thing you can see by looking at this diagram is that this product makes nothing. It is an inactive piece of DNA because it doesn't have a promoter on it. But this one can produce an RNA, and the RNA that it produces is the short little ATTL fragment, which can be seen here. So, you know, because this is a circular piece of DNA, the promoter reads around the construct and produces a little ATTL. We aren't really simulating, you know, rolling circle reading. It would probably read multiple times. That's not part of the simulation. But so this thing right here would produce an RNA, and that RNA is here. This RNA doesn't lead to a protein being made. All it does is get degraded. So RNase is part of this simulation, and what happens there is that you just get your RNA that combines with the RNase, and that leads to it not existing anymore. So that's the degradation process there that you're seeing. On the other hand, this piece of DNA that we started with, it has a promoter pointing towards an RBS and a coding sequence, and that will lead to an RNA that can lead to a protein. So here, for example, because it's a circular construct, actually, it will read around it will read around the circle, and I didn't put a terminator in here, so that's my mistake on that part. And you can see that what you get is an RNA that is too long because it read around the plasmid and went back and made a copy of itself, essentially. That's because there's no terminator in the sequence. Because of that, you'll get an RNA that can make GFP twice in two parts. That's why you're seeing 2 arrows here, or rather you're seeing 4 arrows because sometimes there's not a ribosome bound to the other parts of it. So you can imagine you get a ribosome bound to one of the RBSs or the other, or both of them, and all of that leads to the production of GFP.
I just want to say how awesome I think these plots are, because, you know, you were asking about debugging before. It's very hard to debug complex CRNs, but here you can get little pictorial diagrams to make sure what you think is happening. And I think this is incredibly useful for understanding biological complexity, and it's interactive and automatically generated by Biocompiler, thanks to Andrey.
Yeah, so I find this very useful to, like, you just make sure that the right things are happening to your constructs. Of course, you can also print out the CRN in text form as well. And, you know, if you wanted to know what the text version of all these are, you can have that appearing in these little pop-ups instead of the pictures if you wanted to. You just have to not have the images defined here. That'll happen. So now you can look at the actual text names of the species. If you wanted to, let's say, debug the SVML code, you could look at that. Let's put that back. Right, okay, so I mean, I think that was a lot of like pretty complicated stuff, so hopefully everyone's following here a little bit as we move on to slightly more complicated examples where we're doing the binding as well. Like I said, so here, you know, the integrase mechanism is fairly simplistic. It just goes from the species to the recombined species, and there's no integrase binding. And so you can make a very similar type of reaction where you do have the binding. So here we've defined a new DNA construct that's not circular anymore. And then you can simulate that. And what we've done here is we've defined the attachment sites in a different way. Now integrase binding is true. We don't have to add the mechanism anymore. So last time what we did was we had to add this enzyme integration mechanism because the default is to use the binding. And here I was not using the integrase binding up here in the top cell. But now that we do want to use the integrase binding, we don't have to do that.
So I want to just make a comment here. This is something maybe I didn't do a good job explaining, but in Biocompiler, components can have mechanisms or mixtures can have mechanisms. And say you have a mixture that has a mechanism and a component that has the same mechanism, the component's going to use its own mechanism first before it goes to the mixtures. And similarly, as a user, maybe you have some default mechanism in a component that we've built in our library, and you say, hey, I want to replace it with my own custom version. That's pretty much what Andrey did above. He said, hey, I have this sort of thing that uses this default binding mechanism, but I want to make something simpler, so I'm going to use this new, simpler version, and I'm going to manually override it. And so Biocompiler, as a object-oriented piece of software, lets you do all of that very easily.
Yeah, just to finish up this example here, so now we're simulating integrase binding, and the construct is essentially the same. I mean, I've done it with the reverse site here, kind of like how I did before I changed it. What you can see then when we do this is that now you have all these other species, and these represent the bound version. So now you can see that you have this integrase binding to the attachment sites. And only in cases like this one, where it actually has the integrase bound on both sides, will it actually participate in the integrase reaction that leads to then this one, where it's basically the same thing, except everything is flipped, right? And so, this is another thing that is very useful if you want to model the integrase binding to stuff, in case you care about the fact that, like, in the real system, in real biological system, you can have the integrase binding to the L and R sites, which are not ones that participate in a reaction, but they do bind the integrase. And so, you can model stuff like, sequestration of the integrase by all of these species present in the reaction. You can model that this way. So it really is helpful, to look in here and see how the binding is interacting with your construct. So you can look at that. And you can see also that there are many species that lead to RNA production here. So this is our RNA polymerase, and it reacts with this one to make an RNA. Again, I think we're using a, let's see. Oh, okay, this is what's happening. So this actually, what's happening is that this species, which has the RNA polymerase bound, is participating in this reaction, and the output is that it is now unbound after the RNA is made. So that's what you can see here because of the arrow direction. And here's the unbound version, here's the bound version, etc. Okay, I don't know how much time is left. I think I probably used the entire time.
Yeah, why don't you go through your last example with enumeration, which I just think is kind of cool to see, and then. Oh,
sure. Okay. Yeah. All right. So last example is that now. So previously, in all of these examples, we have had this attribute set, which prevents the plasmid from reacting with itself. Right here. So this attribute is set on any part in the DNA construct, in this case, the promoter, that says that the rest of this DNA construct does not participate in self-reaction. So what we're going to do is we're going to eliminate that, and let's see what happens when we allow the plasmid to react with itself and create all kinds of self-reacted species. So The only thing that we need to do to make that happen is we just redefine the promoter without that attribute. And here we say, this is just defining the attachment sites again, just copy pasted from above. We define our construct the same as before, with the at B site in the left facing direction there. And, you know, just do the same simulation. And so what you can see here is that if you, setting our recursion depth here to 2, so that we don't go crazy, you will see that you get all of these self-recombined species, which are, you know, basically what happens if the plasmid reacts with another copy of itself. You get all these things. And
set the recursion depth to 3 just for fun.
Yeah. So, you know, if you set it to 3, you'll see that there's a lot more things generated, because that just allows the ones that have been generated to react again with all of the other copies that are present in the you know, the mixture, basically, which can lead to really fast, you know, expansion of how many different DNA species are going to exist in your model. So then you have all of these other species that are recombined in various ways. See how long this is going to take. So yeah, likewise, you can plot your network diagram to see what it will look like with all of these added species.
So Andrey, you make Integrase circuits in the lab on a very, very regular basis. Do you see in your experimental work that these higher order reactions matter sometimes?
Yeah, I mean, I think that, you know, it depends on what your copy number of your plasmid is, what, you know, how long do you want to wait, like what's your readout? Because, for example, something like this, this is a plasmid where the promoter either, you know, it either makes GFP or it doesn't, I guess, because it's in the other direction. And if that's your only readout, then, like, you know, you probably don't actually care that much about all of these random DNA things, because it'll still perform the readout function, even if it's in this weird recombined, you know, multimer state, right? Because these promoters, you know, they make GFP or they don't. It'll be the same. However, in this particular construct, what I did is I forgot the terminator, which means that this promoter is going to read through and make, you know, this GFP over here, and that wouldn't happen if it was, you know, in 2 different plasmids. So the fact that it's all one plasmid now is important actually for this construct, because it doesn't have a terminator.
And if you run a gel, would you see some of these like smeared out stuff at the longer lengths?
Yes, I think that you would, absolutely, yeah. I mean, I've definitely seen them in my research, is that you can see these recombined multimers, and they definitely show up on the gel. Yeah, again, the thing is, I mean, if you are interested in just this flipping promoter, you might, you know, put it in the genome or something, because then it's only at 1 copy number, and then you might not see this kind of thing. You might also just do a PCR to check or something across the site, and you might not also see it there either, because the same PCR product could be produced from these. But the presence of these long DNAs could impact your cell's viability. Sometimes cells aren't happy with these super repetitive constructs. They'll try to, you know, repair them in some ways. So, you know, something like this could lead you to an analysis of your construct that leads to understanding why it didn't work or something like that. Like why, you know, like this is just a simple, you know, promoter flip thing, right? Like why would it create this crazy thing? Well, it absolutely would. I mean, because the integrase doesn't care about whether the attachment sites are on the same plasmid or on other plasmids. All it knows is that this sequence recombines with that sequence, and the consequences of that are represented in this model. And so here what you can see is that each one of these are actually like little clumps of reactions. So there's a lot of stuff going on here. What is it? That's it glitching out. Oh, because each time when it generates these, no, it's because the reaction is very long, and so when it tries to appear, it like resizes the whole screen. But then that's funny. Yeah. So, so you can end up with very complicated models from, essentially kind of like a simple starting point, which was just this. And maybe that's something that is interesting to you. Maybe you care about how many of these are made. And maybe you didn't want that and you want to know why it didn't work. And that's something that you can easily do with this sort of setup. Hatem asks, key parameters that control the behavior of integrase circuits. Well, I would say, if you're talking about serine integrase, that is to say, that, at P reacts with at B, et cetera, the key parameters would be the K-int, which is the rate of integration. And that would be the rate at which these integration reactions like this one happen. And you can see here, if you look at the reaction there, that it has K-int there, sort of in the middle of the screen there, is the parameter that it's using to determine that. The binding of the integrase to the DNA is important as well. That's, of course, indicated here in, let's see, where's our parameters? In the KB parameter. So because of the parameter defaulting, everything is defaulted to use the KB parameter unless you specify it to pertain to the integrase specifically by defining it in a slightly different way. And those are the examples that you can see on the GitHub. There's a really good example there for looking at changing parameters like that. Likewise, if you wanted to change the K-ints for specific integrases, it would be very similar. You could just define that here. So I hope that answers your question. And I think that's something also that Ayush will talk about when he does his parameter inference on an integrase model.
Yeah, so we will talk a lot about how the parameters get lumped together and what parameters are important in the reduced models in the next part.
Yeah, what's interesting here is that because this is a RNA that has a lot of promoters on it, it has a lot of reactions that go to promoter things. So that's what we're seeing there.